Hello everyone. Today we are going to discuss about computer-aided drug design and delivery as part of our syllabus in advances in chemistry. So, many drugs obtained from the natural sources are highly valued, but most of the drugs used in the modern medicines are produced from the advances of synthetic chemistry and biotechnology. So, thus a drug can be defined as a substance either by natural or synthetic origins that is used in the diagnosis to cure, relief, treatment, or prevention of disease are intended to affect the structure or function of the body. Thus, a drug is a chemical that affects the body and its process. Normally, traditional drug design is a inventive process of finding new medications based on the knowledge of biological target. It involves the design of molecules that are complementary in shape and charged to biological target with which they interact, therefore will bind. Actually, in traditional approach, uh, pharmaceutical companies develop a synthetic or natural products and they develop almost so many compounds around 10,000 to approximately 10,000 per year. And then out of this 10,000, almost 250 compounds were screened in preclinical studies. Among them, 5 compounds were selected for clinical trials from stages 1 to 4. So, out of 5, only one drug is selected by approved by FDA. That means if you take the ratio 10 in the first step 10,000 active molecules and the last step one drug. So if you cost more than $1 billion and takes 10 to 14 years, means it's a huge investment, huge investment and huge manpower requirement for getting one drug by FDA. Generally, how they do is the rational drug design involves in 5 steps. First one is to identify the cause of the disease. So we should not go randomly to make a drug for for public use. So what we do first, we have to identify the therapeutic area, and suppose I want to develop a antiviral drug or anti-cancer drug. So first we should select the therapeutic area. It should be, if you want to go proceed with the anti-cancer, so we have to focus on the anti-cancer only. Then first we search the target. What is the target? Then we find out the lead compound and to carry out the test for the lead compound. Then clinical trials phase 1, 2, 3, 4, and then approval process. Finally, drug is available. This is the process. So what are the factors contributing for the drug discovery? Advances of molecular biology through output screening and demand fundamentals. These are supply fundamentals. So on hospital is downsizing or disease management, all the global management, all are related to supply fundamentals. Owing to the above factors, regulation, cost effectiveness of drug discovery, and fundamentals of demand and supply, the process of drug discovery is undergoing complete overhaul. Companies that have been making fortune from the sale of drugs are expected to shift their focus to tap into information. So the drug discovery process involves in 2 steps. First one is hit identification. Hit identification involves the screening of large number of compounds to identify those interact with the biological target. In other words, a compound that interacts with the target protein is called hit. So in QSAR, then pharmacophore, mapping, then docking, scoring, de novo, so bioavailability and toxicity studies. After that, it will come. The second one is lead generation. Lead generation involves in the chemical modification of hits by repeated cycles of synthesis and testing of enlacs to produce leads. Leads are the compounds with improved chemical characteristics, thereby increasing suitability of potential drugs. Then another word is lead optimization. Lead optimization involves the further optimization of leads by additional repeated modification of modification to produce drug development candidates with optimized characteristics for further clinical and preclinical development. So computational approaches in drug design. So this is the previous approach, traditional approach. Now the modern approach is like this. First we go with the target selection, then lead identification, lead optimization, finally. So all these things I discussed just now, and anyway, I will go elaborate on this one. So drug designing is selected designed molecule should be organic small molecule, complementary in shape to the target, oppositely charged to the biomolecular target. You should keep in mind these things. Then drug designing, this molecule will be interact with the target to bind the, should interact with the target, bind to the target, activates or inhibits the function of a biomolecule such as a protein. Now the next one, drug design is frequently not necessarily relies on computer modeling techniques. So the type of modeling techniques sometimes referred as computer-aided drug design. This is very much important nowadays. So now I'm going to discuss about the CAD only. So when the disease process is understood at the molecular level, the target molecules are defined. Drugs can be designed specifically to interact with the target in such a way that as to disrupt the disease. Now let's go with the CAD, computer-aided drug design. In computational approaches in drug design, discovery and development process are gaining very rapid exploration, implementations, and administration. So computer-aided drug design is widely used as a new drug design approach to reduce time, cost, and risk-borne factors of traditional approach. So we can reduce the cost of drug discovery and development 50% by using CAD approaches. CAD represents computational methods, resources that are used to facilitate the design and discovery of new therapeutic solutions. What we do in CAD is first we create a molecule, then dock to the target protein, then analyze the molecular interactions, then estimating the binding strength, then estimate drug-like property. So finally, we can get the drug candidate. CAD consists of use of any software program based on the process of establishing standard to relative activity to structure. Computer helps in drug design for storage and retrieval information. For example, structure-based determined experimentally by X-ray crystallography for biological targets like enzymes and drug molecules. And to develop information about the toxicity and structural activity relationship, SAR, computers help is required. To visualize the similarities between and differences between the molecules acting on the same way, and to study the drug receptor interactions, to do calculations involving interaction strength and motion dynamics. Drug design. So with the help of computers, we may use it at any stages of the drug discovery. As I told you earlier, hit identification used by ritual screening, structure-based ligand structure or ligand-based design, hit to lead optimization of affinity selectivity in QSAR, lead optimization of pharmaceutical properties while maintaining the affinity. The main objective of CAD is to change from random screening against disease assays from natural product synthetic chemicals to changing from random screening or against diseases, disease assays, natural product synthetic chemicals to rational drug design and testing, speed up the screening processes, efficient screening, de novo design, target directed, integration of testing into design processes, failed drugs fast, remove hopeless ones as early possible. There are two major types of approaches in computer-aided drug design. The first one is structure-based drug design. The second one is ligand-based drug design. So in CAD, there are two approaches, right? The next one. So these are the two, ligand-based drug design and structure-based drug design. In the ligand-based drug design, LBDD, which is also called LBDD, 3D structure of target protein is not known, but the knowledge of ligands which bind to the desired target is known. So these ligands can be used to develop a pharmacophore model or molecule which possesses all necessary structural features for binding the target active site. Generally, ligand-based techniques are pharmacophore-based approach and quantitative structure-activity relationship, QSAR relationships. So in LBDD, means ligand-based drug design, it is assumed that compounds having similarity in their structure, also having same biological actions and interaction with the target protein. So here a ciprofloxacin structure is there. So this one different size, lipophilic, sp2 hybridization, bonding, all these things are there. So a model of biological target may be built based on the knowledge of what binds to it. This model may turn to be design new molecular entities. Alternately, QSAR, in which correlation between the calculated properties of molecules and their experimental determined from the biological activity may be derived. The QSAR relationship in turns to be predict the activity of new analogs. Next one is structure-based drug design. Structure-based drug design, here in this method, structure of the target protein is known and interaction or bioaffinity of all tested compounds calculate after the process of docking to design a new drug molecule, which shows better interaction with the target protein. The process of SBDD runs with multiple cycles before optimized lead reaches into clinical trials. The first cycle comprises isolation, purification, and structure determination of target protein by following methods. The first one is X-ray crystallography, and then nuclear magnetic resonance spectroscopy, NMR of different types, proton 13 and protein peptide NMRs. We need above 500, 600 megahertz NMR instruments. And then one more model here we use is homology modeling. Using these techniques, compounds comes through the visual screening of databases are placed into selected region of active site of protein. These are the high costly techniques, and it almost above 5 crore or 6 crore rupees it costs, and maintenance also 15 lakhs to 20 lakhs per year. So the from compounds comes from the visual screening are scored and then ranked on the basis of steric, hydrophobic, and electrostatic interactions of these molecules with the active site of target protein. In the second cycle, includes structure determination of the protein in complex with the most optimistic lead of the first cycle, one with minimum micromolar inhibition in vitro shows sites of the compound, which can be optimized for further increment in the potency. So in this, first target identification and validation, then analyze the structure for the protein ligand binding sites, then lead identification and molecular docking, then lead validation and optimization. Finally, we go with the clinical trials. These are the approaches. So after many additional cycles, such as the synthesis of lead and further optimization of lead through the complex structure of proteins and lead with the lead compound, the optimized compounds generally shows marked increment on the target specificity and binding affinity. Already I told you about the homology model, and here in this target, already I discussed about this one. Experimental structure target is not available, possibility to create a homology model of the target-based structure. This one, homology modeling is also known as a comparative modeling of the protein, refers to constructing an atomic resolution model of the target, 3D template. So in SBDD, structure of the biological target and drug candidates are predicted to bind the high affinity and selectivity of the design using interactive graphics, intelligence of medicinal chemists, various automated computational processes may be used to suggest the new drug candidates. So there are some other methods, visual screening. Visual screening is the first method is to identification the ligands for the given receptor by searching the large databases of 3D structures of small molecules to find those fitting into the binding pocket of the receptor using fast approximate docking programs. So it is nowadays it is most convenient tool to find out the most favorable bioactive compounds with the help of information about the protein target or known active ligands. So because of visual screening, the cost effectiveness it becomes more useful in the modern drug discovery. So the types of visual screening approaches are structure-based visual screening, again ligand-based visual screening. In structure-based structure-based visual screening relies on the structure of the target size, protein size, and LBVS, ligand-based visual screening, is based on the estimation of calculated similarity between the active ligands and compounds comes from the database. Now, de novo design of the new ligands. In this method, ligand molecules are built with the constraints of the binding pocket by self-assembling small pieces in a stepwise manner. These pieces can be either individually atoms or molecular fragments. The main advantage of the method is that novel structures can be suggested, means novel molecules can be suggested. Now, third one is optimization of known ligands. So this is can be evaluating by proposed analogs with the binding activity. Binding site identification. Structure-based, this one, binding site identification is also very important. It is in the first step of structure-based drug design. It relies on the identification of concave surfaces on the protein that can accommodate the drug size in molecules that also possess appropriate hotspots, means hydrophobic surfaces, hydrogen bonding, etc., that drive the ligand binding. Then molecular docking is the very important. This one, molecular docking is a in silico method which predicts the placement of small molecules or ligands within the active site of their target protein receptor. Also, the target protein means here receptor. It is mainly used to accurate estimation of most favorable binding models or bioaffinities of ligands with their receptor. Docking attempts to find the best matching between the molecules. It includes finding the right key for the lock. To place ligand small molecule into the binding site of receptor in the small manners, approximate optimal interactions with the receptor. Presently, it is broadly applied to visual screening for the optimization of lead compounds. Here, lead target plus lead equal to molecular docking. So do you evaluate ligand receptor interactions in a way that may discriminate the experimental observed mode from the others to estimate the binding affinity? Now, important. The components of docking is first one, pre and during the docking, representation of receptor binding site and ligand. The second one is during docking, sampling of configuration space of the ligand receptor complex. Third one, during docking and scoring, evaluation of receptor interactions. Now, major success stories of CAD is K plus ion channel blocker, structure-based drug discovery, Ca plus 2, calcium plus 2 antagonist, P channel blocker, chemical descriptor-based discovery. The major applications of computer-aided drug design is cost-effective, time-saving, rapid, automatic process. It gives reduction in the synthetic and biological testing efforts. It gives the most promising drug candidate by eliminating the compounds with undesirable properties like poor efficient efficacy, absorption, distribution, metabolism, excretion, and toxicity, etc. through silico filters. We can know that the drug receptor interaction pattern through computer-aided drug design. It minimizes the chance of failures in the final phase, which saves a lot of time and money for pharmaceutical companies. So by this, I have discussed all the major points of computer-aided drug design. Thank you.